All right. I was gonna say this is gonna be a terrible recording. It's just me going doody-doo. So, all right, let's get our screen shared here. Yes. Did we get into this? How far did we go? Oh, come on. In the butt. Okay. Okay. Yeah, we talked about this a little bit. So when we left off, we had been talking about the globins. We have our myoglobin and our hemoglobin. And the big thing that we're looking at was the ability of them. Let me go back up here. We have this coordinated iron atom in the center of our porphyrin ring. So the iron is coordinated by 4 nitrogens within that ring. There's a 5th nitrogen from a histidine, and then there's a spot open there for it to bind oxygen or carbon dioxide. Okay, so we're gonna look at this, and I don't know if you're talking about this class being the hard class. Is this the hard one? Yeah, yeah.
It's hard because the materials.
Yes, exactly. Yeah. I was gonna say, individually, I don't think there's anything that's really super challenging to understand in this class. The hard part is there's so much information, and then everything affects everything else, and everything is interrelated. So it takes some time out of class to get it to sit in your brain. At least for me, and I always mention when I studied for CUMES after my first year, I started a good month in advance, and I just start writing stuff down. I'd study like 14 hours a day for the first several days, and then it was like 12 hours a day, and then 10 hours a day, and then by the day before the exam, I was like, all right, I'm gonna spend 3 hours and look over this. But that was high end, high motivation. This is my job. I got nothing else to do, type of deal. So I try, and we don't go into that much depth as undergrads. Okay. So again, we're looking at our porphyrin ring that's inside. That is our prosthetic group for both myoglobin and hemoglobin. Okay. If we look at the heme binding, we can see this effect of carbon dioxide versus oxygen binding. So what we have are a few other structures in here that kind of reduce the favorability of binding carbon monoxide and enhance they don't really enhance, but they make it harder for CO to get into the binding pocket. Still, carbon monoxide is much better at binding. It's like 200 times better than oxygen. So as long as we're not in a carbon monoxide rich environment, we're okay. So as the temperatures drop, don't start your car in a garage or something. Yeah. Always, the self-censor. Basically, every time you hear flying up there, I'm like, okay, I'm not gonna say that. I'm not gonna say that. So, but yeah, I like this slide just because of this picture on here. We get to kind of see some three-dimensional structure in here. All right, we're coordinating our iron. We have our bound substrate, our ligand, I should say, our ligand. And then we help coordinate that ligand in there. This hydrogen on this distal histidine from that E7 alpha helix helps hold everything into place. If you happen to go into molecular modeling of biomolecules, you start to get used to this stuff. But when I talk about like the E7 chain in here, if I go back up just a little bit here, we're really kind of talking about these different sections here. So here's our histidine 64 that comes over this way, maybe sneaks in there and helps stabilize that oxygen in there, okay? And helps make carbon monoxide a little harder to bind in there. Okay. I think I had mentioned, how do you treat carbon monoxide poisoning? You just give them oxygen, lots of oxygen, so you can outcompete it. Yeah, you just want to make sure that your partial pressure of oxygen is thousands of times greater than your carbon monoxide. Contrast that with cyanide. Cyanide binds irreversibly. In chapter 6, when we start talking about inhibitors, we would, kind of use the analogy that cyanide would be like a suicide inhibitor. It's basically going to covalently bind to that heme group and essentially take that whole porphyrin ring, that whole heme, out of the equation so that it can't transport oxygen. So the treatment for that is like blood transfusion and cyanide binding.
For Eva?
Yes.
Can you check my messages?
Uh-oh. Okay, yes. Give me just a second here. Messages in the chat here, or how do I get in there? Let's see here.
Yeah, and Zoom.
Where are we? You gotta see my right view. There we go. Okay. Where is that showing up here? Come on. Okay, yeah, I can see that in there. Okay. I think that too. Cool. Yeah. So much for the private messenger function. All right. So we're good there. Sharing screen. Okay, let's get back to this. Let's take this and shrink it down. Get back here. There we go. I really need my multi-monitor setup. All right. Okay. All good? Okay. Okay. So yeah, we're essentially looking at this kind of 3D structure and trying to relate that structure to the actual function and purpose of our protein. Okay, and we're going to be doing that this chapter and the next chapter, which is the scary one. The next chapter is the one that makes everybody think biochem's hard. It's just got to be systematic. Okay, so let's talk about our oxygen binding to our various globin proteins. All right, because of all those conjugated ring structures, we end up having a very strong chromophore, so we can see that oxygen binding very well. We would see this at 429 nanometers. Do you need to know that? No, I'm not going to be that picky about stuff like that. If I were making like a full multiple choice, let's weed out everything, every possible question, that would be crazy in there. But it is one of the kind of the neat things where you can see like high oxygen content. If you've ever done like the, go to like the hospital or doctor and they put a little thingy on your finger to monitor your oxygen saturation. That's essentially what we're doing here. If you look at the blood in your arms, you can see your veins are kind of purplish, and of course fresh blood is very red. And we basically see that color shift based on the amount of oxygen bound. So if like cut your vein that's purplish in there, as soon as that blood hits the air, the oxygen will bind to the heme, and that's why blood is red. Okay. Anybody know what color crab blood is? It's kind of greenish, right? So we have like red purple for our hemoglobin. Crabs will also have hemoglobin, and other crustaceans, and I think there's several other animals that do this as well too. Any idea why their colors are kind of greenish as opposed to red purple? So trivia, instead of using an iron-based heme to transport oxygen in their bloodstreams, they use a copper-based heme. So that copper tends to give it a greenish hue. So if we go back to like mythology, we could say like demons with green ichor. They must have copper-based heme. So anyway, random stuff in there. But we can monitor this oxygen transport. Okay, so we sort of saw, or this level of oxygen binding, we sort of saw this. We saw this binding curve before. At very low pressure, we saturate our myoglobin. So myoglobin is fantastic at binding oxygen. So the question we have here is, can we use myoglobin to transport oxygen? Okay. All right, well, let's take a look at our data. So the partial pressure of oxygen in the lungs is typically about 13 kilopascals, right? Which, doesn't mean a whole lot to me because I'm like, oh, what's the pressure in American units? All right. But basically we have a very high concentration of oxygen in there. It's off the chart here. So if we have myoglobin in the lungs, it's going to be just saturated with oxygen, okay? We get to our tissues, okay, and our oxygen concentration changes. We've only got about 4 kilopascals of pressure. That's way about here. But the trouble is, with myoglobin, that's not a big change from there. Myoglobin does a fantastic job of binding oxygen. So good that it doesn't want to give it up. I like to think of like the seagulls in Finding Nemo and Finding Dory, and it's like, mine, 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 mine. That's my attempt at humor. It's like, oh, it's not very good, Ducky. All right. But you have to get down to a partial pressure of oxygen well below 1 kilopascal before myoglobin is going to give up that oxygen. Okay. So myoglobin does not make a good transport of oxygen. Right. It's great at storing it. You can put your oxygen there and it's going to hold on to it until you're severely oxygen depleted, like high anaerobic activity. Say like you're cycling up a mountain in a Tour de France. I don't know why I watch the Tour de France. I have no interest in cycling. I just like to watch people struggle. That's why I'm a professor too, right? Oh, okay. Dark humor. Okay. So the crazy thing is, this is our myoglobin curve right here. It's, on this scale, you can't even barely see it. It looks like the border's in there. So, myoglobin, great for storage. But for transport, we need something that's going to release it, okay? You wouldn't want to like load something on the truck and then have it never get off the truck. So what we see is typically our lung pressure over here in kind of pinkish and our tissue pressure over here in blue. So we have to use something else that can bind oxygen really well in the lungs and then release it in the tissues. And we have another trick. So you can see here we have something called a high affinity state and a low affinity state, okay? So this is gonna be our same protein, but we're going to have it interact in such a way that when it's in the lungs, it's really good at binding oxygen. You get to the tissues and it shifts to a different form and it releases that oxygen. Still gonna bind a little bit, but it's gonna release it far, far better than myoglobin would, okay? All right, so what we have going on here is an interaction between multiple binding sites, okay? So we need a protein that is going to be able to bind multiple molecules of oxygen, right? And these different binding sites have to interact with each other. They have to communicate and coordinate. So we call this cooperativity, right? So we can have, two types of cooperativity. We can have positive cooperativity, where if one site binds an oxygen, it makes the other sites more likely to bind it. Okay, that's gonna be positive. We can also have negative cooperativity, where if we fill in one binding site, it makes it harder to bind at the others. In our case, we're gonna be talking about hemoglobin, and that's gonna be an example of positive cooperativity. It's kind of like peer pressure. It's like, if your friends were smoking, would you start smoking too? Whatever, I don't know. It's my dad voice there. The neat thing about positive cooperativity is we're going to have this sigmoidal binding curve. So if I go back up to that previous slide, you can see this sigmoidal binding curve as opposed to the hyperbolic for our single state. Our sigmoidal says, hey, we've got some stuff that's cooperating, all right? And we'll talk about a hill plot here in a minute, okay? So let's look at this in kind of our cartoon form a little bit, okay? I always debate about how helpful is this picture, but what we've got going on here is we have, our binding site over here that can kind of flex around, and here's a binding site on the other side. So what happens is when we bind a ligand over here, okay, it's going to induce a conformational change to make, in this case, for positive cooperativity, it's going to induce this conformational change to make this binding site more attractive, give it higher specificity for a second ligand that can bind there. So that would be our positive cooperativity. If we had negative cooperativity, when we bound here, this one would get all crazy and make it harder to bind. But we're mainly going to talk about positive cooperativity. Okay. So you guys been driving around town? Anybody see like Halloween decorations up already? Yeah, one of my neighbors has like ghosts hanging out. And then there's like some just general fall Thanksgiving stuff. But with Halloween coming, here's a scary slide. Don't worry, we're not going to do the math. Okay? We can look at this quantitatively. Okay, so I'm just going to kind of go over this to just look at it. We can look at the effects that our association constant is going to have on here. So if we have multiple binding sites, we essentially have N is going to be our number of binding sites. So if we've got more than one, N might be 2, 3, or in the case of hemoglobin, 4. Okay, so our theta, then we have to start putting in some exponents in here. Okay, so we can see this big change. All right, if we take the log of both sides, we end up with this crazy looking expression, something like this. Okay, now the key to all this math that we're not going to go into is looking at that value N. If N equals 1, that essentially says each binding site has no other effect on the others. So we have no cooperativity. We would see our standard hyperbolic curve. If we have multiple binding sites and they don't interact, our N is going to equal 1. If our N is greater than 1, we're going to see positive cooperativity. So a greater than 1 value means the multiple binding sites are interacting with each other and making it more likely to bind when something else binds. Okay? And if we have an N of less than 1, that would be our negative cooperativity. We're making it more difficult. Okay? So I'll use an analogy that is, annoying in real life. So, for example, I had the lab I joined had a senior grad student in it when I first came in, and he was quite good at his job, and so he went off and got his career. It was all well and good. Then I had a friend a couple years behind me who was from the same country as this guy, and I was like, hey, maybe you could put in a good word or something like that, or just let him know about job openings that were coming up, and stuff like that. But he never did. In fact, actually, he made it harder for my friend to get a job. So that would be negative cooperativity. On the other hand, if, a lot of times when people do job searches, they'll network and they'll make friends with people and say, hey, I know this person is really good. That might be an example of positive cooperativity. Hey, we need another person here. I know someone, so go ahead and bring them in. I don't know if you guys like that analogy or not, but that sort of helps explain our cooperativity. Okay. If we take all this math and graph it, because a picture is worth 1,000 words, okay, we can see something cool here. So in our case of myoglobin only has 1 heme in it, so it's only got 1 binding site. So when we plot all this stuff, our log of partial pressure versus our log of binding sites, you're good. You'll get a nice straight line in there, okay? So this is basically taking our hyperbolic curve for those binding pressures and making a log plot of it, and you get a straight line because there's no cooperativity. Our slope is 1, okay? If you have positive cooperativity, like with hemoglobin, where the 4 binding sites interact with each other, what you'll see is this sigmoidal curve will show up here in our log plot, and you can look at the slope of the sigmoidal part, okay, which ends up being somewhere between 2.8 and 3.4 usually. But what this means is that we have multiple binding sites that are interacting with each other and for cooperative binding. They're making it easier. Once one oxygen molecule binds, they make it easier for the next one. So it's kind of like once one person gets hired, they make it easier for the next 2 or 3 people to come in and get hired too. Okay. Yes?
So is it the same backwards, like once you let go of one oxygen? I mean, never like that.
Yes, it does the reverse as well too. So when you have this conformational change as you release an oxygen molecule, that's going to influence the other sites to change as well to release it. So even though that's kind of a release, I tend to think of it as being negative, it's still positive cooperativity because all the sites are starting to line up and do the same thing. So they're either all going to be in the high affinity state where they all want to bind, or they're all going to shift to the low affinity state. Okay. So you ready? What I just said was really a lie, okay? Because they don't all suddenly shift one way or the other. It's a process, okay? And there's 2 ways this can happen. We could go from, all in the high affinity to all in the low affinity. So we have our ligand bound in the end. Once we have one, maybe we shift over this, or maybe we don't, okay? That would be great if it was kind of binary and we're all high affinity or we're all low affinity. But realistically, we can have like one position in low and 1 in high affinity and 3 in low affinity, but then they all shift. So we get a second one over here and a third one and fourth one. You know, but as we bind, we make it easier and easier for this to happen. So I don't really like this slide very much because this is one that I think you kind of have to stare at and say, what in the hell are they saying here? You know? Or in this case, what in the L are they saying here? For our ligands. Oh, I'm hilarious. Okay. So, I believe so, yes. That's all we got here. So it means you're all low or all high. So I think, yeah, in the boxes they're distinguishing it as high. So note, even in low affinity, you can have ligand bound, and even high affinity, you can have ligand bound. It'll depend on the partial pressure of the oxygen. You can have everything in low affinity and still have the molecule completely saturated if you just have that much oxygen there. So remember, our ligand bound state only depends on concentration of the ligand. Okay. But we're just more likely to bind if we're in the high affinity state. Okay. And we're more likely to release if we're in the low affinity state. Okay. So the universe is all statistics. All statistics. Okay. Okay. You guys with me so far? Cooperativity is like, all right, yeah. It's kind of weird lecturing this way because I can't see any of my online people, so you know, I really do need a second monitor. Okay. So cooperativity ends up being a special case of allosteric regulation. So if we go back and look at just that three-dimensional structure of the binding pocket for oxygen, remember you've got that histidine coming in there that makes it harder for carbon monoxide to get in there. Well, our cooperativity is similar to that. It's just this cool little special case, okay? So we change the conformation, or we have a conformational environment that can either promote or inhibit, in this case, binding, okay? All right. We could also talk about, you know, in terms of our allosteric regulation, we could classify it as homotropic and heterotropic, where, you know, the same ligand, when it binds, it makes it more likely to bind more of that ligand. You could also have different ligands that bind in there as well too. And we'll see an example of that with our 2,3-bisphosphoglycerate coming up, okay? But cooperativity is positive homotropic regulation. We have our ligand bound. It induces a conformational change that makes it more likely to bind more of that ligand at other sites, okay? I think this particular concept also helps with the induced fit model as well too. So everything is very dynamic. There's a lot of flux and change that's going on. So as we bring in our ligand, right, we go back to just our simple one protein, one ligand, you know, we have a particular shape and our ligand comes in here and we have this induced fit where the protein changes its shape to bind that ligand, and then it can change back to release it, okay? So we've got the same kind of deal going on here. We have one oxygen coming in, and it changes this conformation to make this more likely to bind, but as it changes the conformation here, it travels all the way to a different section of the protein and changes this conformation. So a second one is more likely to bind at a different site. Okay. Good times, right? Yeah. That's why I like biochem. It's fun. All right. Okay. So this has all been kind of general. I like this chapter because we can be really specific. We can look at hemoglobin, and we're going to compare myoglobin and hemoglobin. If we look at, you know, myoglobin's 3D structure in a ribbon form here, and we compare that to a beta subunit of hemoglobin, all right, you can see that the structures are really similar. You know, you got this nice little, I think this would be the E chain in myoglobin. There's a similar one in this hemoglobin subunit. You can see a little shorter alpha helix over here. Same way, this one's longer, a little shorter over here. We got a little longer one here, but the overall three-dimensional structure is very similar. Okay, it's kind of like the difference between like a Chevy pickup and a Ford pickup. It's probably like heresy for somebody. But you know, they're both pickups. They both are going to hold stuff. They're just built, shaped slightly differently. You go back to like World War II on the West Coast, and you have spotters out there, and you look for the shape of the airplanes to identify them. All right. So if we look at hemoglobin, we actually have 4 different subunits that come together. We talk about mature human hemoglobin, you've got 2 alpha subunits and 2 beta subunits. And then there's a little region where these subunits interact with each other. Okay? So if we bind an oxygen down here, we're going to see this conformational change in one of these arms to the subunit. And that arm is going to interact with the arm of a different subunit. And that's how the cooperativity actually works. If we try and look at this in like super detail, I'm going to blow this up, man, look in here. So we have this particular histidine. You guys do not need to know this level of detail, but you know, this histidine is going to interact with this lysine on the other chain here and just shift it ever so slightly, okay? So it's kind of like if you're standing on one leg and you're just going to shift over to the other leg. Well, that subtle little shift is enough to make it more likely that something's going to happen. In this case, we're going to bind more oxygen, or if we shift the other way, we're going to release the oxygen that we do have bound. Okay. If we want to map this out, I'm not going to make you guys do this. We could actually, you know, look at these interactions in here and see what's actually happening in there. All right, but let's not sweat that too much. The key is, you know, we're just good understanding of cooperativity. Okay. Maybe a sample question. Explain cooperativity in detail. It might be a question with the graph, or it might be, you know, it might be a factor in a large question, like, you know, compare and contrast hemoglobin and myoglobin. Okay. So, and one of the big things there too is myoglobin only has 1 subunit. Hemoglobin has 4 subunits. Because myoglobin just has the 1 subunit with the 1 porphyrin ring, it has no cooperativity. It's only got 1 binding site. Hemoglobin is different with 4 subunits that interact with each other. It can exhibit cooperativity so that those binding sites can talk, so that we can have a transition between a high affinity and a low affinity state for oxygen. So thus, myoglobin is a great storage protein for oxygen because it's going to bind it and hold on to it until you have really low partial pressures of oxygen. Hemoglobin makes a good transport because it can bind it in high concentrations, move to areas of low concentration, shift its conformation to be less, have a lower affinity for oxygen, and release the oxygen there, and then go back and pick up more. Yes, yeah. So if you go into like the tissues or the muscles or something like that, the hemoglobin, so hemoglobin will be in its high affinity state in the lungs, move through the bloodstream to the tissues, shift to the low affinity state, release oxygen, and come back to the lungs, shift and bind more again. Okay. So this is a terrible picture because it's really tiny, but the key here is we're looking at protein homologies. We're comparing myoglobin and hemoglobin, and we see there's a lot of similarities. There are a lot of conserved residues in there, and even like semi-conservative things as well too. For example, if you change like a valine to an isovaline, it's roughly the same size, it's the same hydrophobic character, so you're going to get probably a similar overall 3D structure. If you change like a valine to an arginine, suddenly you've got a very different size, and go from uncharged to charge. So that would be rough. So these just basically look at levels of homology between the 2 different subunits. And the idea is the sequences for both hemoglobin and myoglobin are very similar. Okay. All right. So let's take a closer look at hemoglobin. All right. I have random stories that I like to tell as like a nice little break, but we'll get through this little section and we'll do a break. All right. So our hemoglobin, we've already mentioned this high affinity and low affinity. We make it worse by defining them as the R and T states. Okay, so the T is for the tense state, all right, and the R is for the relaxed state. So the relaxed state has a greater affinity for oxygen. How do you remember this? Here's how I remember it. What happens when you're tense? You're close like this, and you grind your teeth, and you don't take a breath, right? And then someone says, relax, okay, and you take a big breath. Take a big breath, you get more oxygen. So the relaxed state is higher affinity. I would think if you're binding something, that would be a little bit more tense, but it took me a while to figure this out. So that was my little mnemonic for remembering that relaxed state has a higher affinity for oxygen, tense state has a lower affinity. So we go into the lungs, take a big breath and relax. We bind our oxygen. We travel to the tissues. We get tense. We release the oxygen. And then we repeat. Okay. All right. And again, if we go back to that interface on those previous slides, or if we take a look at it here, these alpha beta interaction in the arms in here their ion pairs are going to induce that conformational change. Okay. So we know that the different subunits talk to each other. How do they do that? So when oxygen binds, we're going to see this shift in this leucine down here, and this leucine and this valine up here. So oxygen binds, those make a little subtle shift. That's going to carry over into those arms that are going to influence the next ones. So oxygen, the conformational change is triggered when oxygen binds to the iron and the heme in there. And we get this subtle little shift, which is enough to go from the tense state to the relaxed state. Let's say don't overthink. Okay. All right, forget that next slide. Yeah, woohoo, back up. Pause a moment. Okay, so we're good there. We got cooperativity. Basically, oxygen binds. It makes it more likely that the other three sites are going to bind. Okay. Right. Oxygen releases, and we get the reverse thing. So if I release my oxygen, we're going to see this shift back this way, and we're going to go to the tense state. Okay. All right. So we've got our allosteric regulation with cooperativity. Guess what else we can do? pH can affect the binding of oxygen as well too. So if we change the pH, it's like, okay, changing pH, we're going to change some function there. We see that our binding curve changes. All right. So the higher the pH, the stronger the affinity for oxygen. Okay. So I think this is one of the harder things to, we're increasing our pH. We're making it more basic. If we're more basic, we're going to bind oxygen more strongly. Okay. All right. You can maybe, I don't know if this is a good mnemonic or not, but I think what happens when you work out, you use a lot of energy, you get lactic acid that builds up in your muscles, right? So you're increasing acid. So if you're increasing the acid, then you're decreasing the affinity for oxygen. That's why you gasp for breath after working out. Climbing up the stairs, I need more oxygen because I'm in a lower affinity state with the more acidic. It doesn't quite work that way, but that might help you remember it. Okay. So if we increase the acidity, we're going to decrease our dissociation. Yes, decrease our dissociation constant. Okay. So let's look at this. All right. So actively metabolizing tissues are going to generate protons. Right? We're making lactic acid. Right? So, and other acids as well too there. So we're going to lower the blood pH near the tissues. All right. So we're going to end up making our bicarbonate in there. We have CO2 that's being produced. We'll make more of that in there. So our affinity of hemoglobin for oxygen is going to depend on that. Our proton can bind to hemoglobin and stabilize that tense state. Okay, so it's going to lower the affinity for oxygen. Okay. One neat thing about this is in muscles where you're producing this lactic acid and you see that increase in proton concentration, that decrease in pH, you're going to statistically make it more likely that you're going to release more oxygen so that you can continue to undergo oxidative phosphorylation and metabolism. Okay. All right. The other neat thing is if our proton binds to our hemoglobin, it can get carried back to the lungs. So you can look at this pH difference between the lungs and the tissues. That's going to help increase the efficiency in here. This effect of pH on the oxygen binding is known as the Bohr effect, primarily because if I lecture about this for a whole long period of time, everybody will fall asleep. Because it's so boring. All right. That's why I'm a professor and not a comedian. Okay. Actually named after Niels Bohr. But anyway, we change the pH, we change our binding affinity. So we've got cooperativity, And we've got pH, right? And of course, tissue-specific pHs as well too. All right, let's talk about getting rid of CO2. We tend to think most of our CO2 is being bicarbonate, or sodium bicarbonate in the bloodstream. But we can also remove a significant portion of the carbon dioxide that's generated in oxidative phosphorylation by making a carbamate on the end of one of our amino terminal residues of these polypeptide subunits in hemoglobin. Okay? So we can carry this to the lungs, then we regenerate CO2 in the lungs and exhale it away. Okay? So the other neat thing about this is we generate a proton. Oh my goodness, our proton can bind there, so we're going to contribute to the Bohr effect when we have CO2 bind. So the carbamate gives you additional salt bridges, which is going to help stabilize that T state as well too. All right, so it's like German engineering. Joke about that for me. All right, everything's kind of fitting together to promote the T state in low concentration oxygen tissues and the R state in the lungs. All right, you ready for some heterotropic regulation? Now I feel like I should be doing an infomercial here, but wait, that's not all. It slices, it dices, it juliennes. We also have another molecule that can bind in our hemoglobin. It's got a particular cavity in there that it will bind to, and it can affect the affinity of hemoglobin for oxygen too. All right. So our 2,3-bisphosphoglycerate has a nice little binding pocket in there. It stabilizes the T states. Okay. All right. We can call this 2,3-BPG for short. If you take Biochem 2, we'll talk about 2,3-BPG and 1,3-BPG. All right, but it's a small negatively charged molecule. You don't need to know the structure. We're good on that. It binds to the positively charged central cavity of hemoglobin. Okay. All right. So let me ask you a question. So we're all down here relatively close to sea level. But suppose, we take like a 2-week trip and go to Colorado up in Denver, so we'll go to Denver, the Mile High City, and we'll do some learning there, right? Okay. What's going to happen to, your lungs? Yep, you're in a higher altitude, right? Yes, because there is less oxygen in the atmosphere the farther up you go, so it's going to be harder to do that. So what do you think would happen to BPG levels? It would increase. It would actually increase because what's going to happen is at higher levels, you want to be able to release that oxygen in there. So you're going to see BPG increase so that it will promote the release of oxygen in your body, even though there's like a lower oxygen level out there. So we'll see that 2,3-BPG levels increase at altitude training. Yeah, so since this is going to stabilize the T state, it's going to inhibit oxygen binding in the tissues. So basically your partial pressure of oxygen are going to drop. We make more 2,3-BPG to make sure that the oxygen that is carried in there gets released in the tissues. And of course, our 2,3-BPG can unbind a bit as you get into the lungs to help promote it. Okay, if we look at this pictorially, our BPG is going to bind in here. Here's our heme, so this is kind of far away, but our BPG can bind in the center of the hemoglobin and promote it. And I know you guys just look at these two pictures and say, oh yeah, they're obviously different, right? Yeah, you ever do those? What's different between these two pictures? Well, yeah, this is our R state and this is our T state. So basically, our R state, we kind of close up that cavity a little bit. Our T state, and we have a space in there for it. Okay, so this will be our altitude adaptation. Okay, so you can actually monitor the BPG levels. If you look at this, if you had no BPG, your hemoglobin curve would look a lot like myoglobin. So typically what we'll see at the partial pressure of oxygen in the atmosphere at sea level and at altitude, okay, is going to vary. So we will vary our levels of BPG to accommodate. just to kind of shift this curve down a little bit. There we go. All right, awesome. Good times, right? All right, that's a lot. Okay. What else did I talk about here? Just to go on a side little bunny trail. So like I say, I'll watch the Tour de France every so often because it's on in the mornings when I wake up and I'm like, oh, yay, bicycles, and they crash and they go real fast and, they're really whiny and they dope a lot, right? So the big thing was a couple of years ago, there were several riders from like Colombia, Peru, South America, and up in the Andes Mountains that were doing really well because they're born at altitude, they train at altitude. So, when they're doing these mountain rides in France, they may have a slight advantage for being able to bring in oxygen and have all that good stuff going on there. Of course, what's the big problem with the Tour de France? I don't know if you guys would even hear about this. Doping, right? Performance enhancing drugs. So if you go back to the start of the Tour de France in the 1800s, I mean, we have bike riders taking cocaine on the first one. Let's go. But it's been this huge arms race of who can dope the best. Look at Lance Armstrong with 7 Tour wins, and he doped to be able to do that. I actually read a little thing about the first American to win it, Greg LeMond. I mentioned that because I had an awesome Halloween costume last year, but we'll get to that when we get closer to Halloween. I'll share that. But he noticed on one year when he was in the Tour, like he was one of the best cyclists in there. 2 years later, like the people who were okay cyclists were suddenly all passing him. So sometime in the 80s, the doping really took off. And then you get into some crazy stuff like erythropoietin and how to elevate different levels in there and how to enhance your ability to bring in oxygen, get it to the tissues, release it, burn off lactic acid. There are so many little metabolic factors in there that you could target to improve athletic performance. And part of me is cynical and says, well, why even bother testing? We'll just let people go crazy. You could have like a dirty Olympics and a clean Olympics. So you can look at like the Russians in the Olympics who were banned from, was it the Summer Olympics a couple years ago, because so many of their teams, pretty much the entire weightlifting team tested positive, and it's great. And you see the same thing in like the NFL and Major League Baseball. Performance enhancing drugs are a big issue. Should they be? Well, it looks at the health effects, and prolonged usage of a lot of different classes of PEDs tend to have negative effects. We talk about steroids. That's probably the most common one for building bulk muscle. Steroids are prescribed a lot for, like, say, if anybody ever has an asthma inhaler, it's a steroidal, and they're to increase the muscle contraction of the lungs or something. So you can basically breathe better. Okay, so it treats a condition in there. I know a couple years ago we looked at tennis players. There's some, Russian tennis player, and she was banned from the tour because she was taking some medication that was, it had a use for maybe like treating asthma, but, turns out apparently all the Russian tennis players had asthma or something, because it also had another effect in there, an off-label usage that could enhance performance in theory. So this becomes something ethically to look at. Suppose you have kids and you come from a short family, you want your kids to be like basketball players, you can give them human growth hormone, right? Or you can enhance them, right? So those are things that we're going to have to figure out how much body modification is okay. All right. So of course, I don't think we could actually get through hemoglobin without talking about sickle cell anemia. Sickle cell anemia and sickle cell trait and sickle cell disease are due to a mutation in the hemoglobin. There's actually a glutamate that gets converted to a valine. All right. So what this ends up doing is this valine side chain now can bind to a different hemoglobin molecule, and you end up with something. We go back to like those misfolded proteins where you could see you got those different beta sheets that could start lining up to make amyloid plaques. This valine essentially allows that to happen with the red blood cells. So instead of having this circular corpuscle, you end up with this sickle-shaped one in here. Homozygous, not good. You can't transport oxygen, can't bind and transport oxygen very well with homozygous sickle cell. But heterozygous is usually a positive trait because it is a resistance to malaria. So again, you can see this in populations that come from Africa, where there tended to be higher levels of malaria, are at a greater risk for sickle cell trait. I believe, I was thinking like it was the Pittsburgh Steelers had Ryan Clark who had sickle cell trait. So he wouldn't, speaking of Denver, he wouldn't go play in Denver when they went to Denver because the low oxygen levels could actually trigger a sickle cell flare-up in there, which could be life-threatening, blood clots, all that good stuff. So you can see, basically you can have multiple hemoglobins start binding to each other, and then you end up with these little strands, and you get these fibers, and the amyloid plaque, which ends up potentially being a blood clot and being deadly. All right. Okay. All right. So that's pretty much everything I want to say about hemoglobin and myoglobin. I think hemoglobin and myoglobin is a great case study. We talked a lot about it. We talked about lots of ways that we can affect the binding of oxygen to hemoglobin. You've got cooperativity, you've got 2,3-BPG, you've got altering the pH. So, lots of juicy questions there. So knowing hemoglobin and myoglobin pretty well is probably a good thing for the upcoming exam. It won't be for a while though, probably not until the first week of October. Yeah, lots in there. So the rest of this chapter, we can kind of break down into talking about antibodies and antigen interactions. Again, these are proteins and their ligands. And then we're going to talk about actin and myosin a little bit as well too. So let's see about covering, actually, nope, that's right. I forgot. I wanted to go over the exam, didn't I? We'll pick up with this next week. Yeah, I keep thinking, still, by the end of the semester, I'll get used to the fact that we're now only 2 days a week instead of 4 days a week. All right, so let's look at the exam. Again, I'm the typewriter from Sesame Street. Okay. Where's that? Let's go back here. My dashboard. Back in one. Okay. So I think you guys can all see the answers you gave. Is that correct? But it doesn't show you the correct answers for the multiple choice ones yet too. I'm going to see if I can go ahead and change that right now. This is under assignments, isn't it? There we go, exam one. Let's go in here and edit this. Okay. When I was a young new professor, I put in lots of feedback in the questions. So, yeah, but, all right, now what we have here. Let students see the correct answers. Okay. Oh yeah, I can actually do this now. Okay, so done. And just in case, we will go to, because I'll forget for the next time we do this. All right, there we go. Woo. Okay. Access code, never good. Okay. All right. So the good news is now you'll be able to go back and kind of look at the multiple choice ones. If I actually click in on this, preview it, kind of go through a couple here. So I think I'm trying to think if there were any particular questions that gave a lot of people a lot of trouble. The good news is there's kind of like a nice mixture here. I'd say the autotroph one was maybe, a little challenging. Okay. That's back. There we go. Yeah. See out there. So even though I'd say with question 9, even though I don't give you any of the pKa value for this, if you look at some extreme like this, like a pH of less than 2 or greater than 11, you can pretty much assume you're going to be all acidic or all basic form. So in this case, in its most basic form, we're going to strip off all the protons we can, which is going to be B for that. You guys like that? Yeah, I figured that was the easier way. I figured out, so in my answers, I can't put superscripts and subscripts, and it just turns into a mess if I try and put all the little stuff in there. So I did it this way. All right, conjugate prosthetic group, yada, 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 not too bad there. Okay.
Dr. Ebert,
Yes.
the share screen froze, so we can't see what's going on.
Ah, okay. Let me try that again there. Is that better?
Yes, we can
Yes,
see.
sir.
Okay, good. All right. Question 14, it looks like there was something funky there where you could actually choose multiple answers. So maybe I said the wrong, but I just wanted the one best one. And in this term, it would be, we have glycine, valine, and serine. So glycyl, valyl, serine is going to be our actual correct answer. In this case, the first one, glycyl, valyl, serine.
So I picked two. I picked that one.
Uh-huh.
I got it wrong.
Yeah. Or you picked what? You picked this one and got it wrong?
Yeah.
Okay. Shoot me an email and I'll take a look at it because, like I say, I think people could pick.
No, I picked the one that has dashes between.
Yes.
That one it was two choice and multiple choice.
Yeah, I picked only that one. You
Okay.
remember when I asked you how many questions?
Yeah, it should be. Okay.
Yeah, and it says wrong.
Okay. I'll go back and look at that just to make sure because I like I say, I know a couple people did. Yeah, so I'll make sure this is question 14. I'll take a closer look at that one in there. All right, that was my parallel question there. Okay. We got to question 16 and, not too bad. I think there were something like 7 distinguishing characteristics of living versus non-living organisms. I wasn't looking for all of them, but I was looking for 4 or 5 of them. That would be good. Some people just gave me 1 or 2. So I'm like, I'll take off a point or 2 here. And then the other thing, looking for the specific example of a biochemical process. Some people gave me really detailed examples in there of like glucose metabolism or something like that. But I was looking at something in particular, maybe say ATP generation would have worked, but any particular example that would work. For ATP, we're extracting and creating energy from metabolites taken from the environment. You could look at DNA and say, well, we're transmitting genetic information to future generations in there. That would work. Okay, but generally this one wasn't too bad. It's just kind of completeness. I think you guys were generally fairly prepared for equilibrium versus a dynamic steady state. Some people would actually went into a little bit of energetics and talked about the thermodynamics behind it, so that was good. The big thing, and there are a few people who were just a little confused on that, but overall, I think question 17 was fairly strong for the class. Big thing here is our equilibrium, the rate between reversible reaction of reactants to products is flowing in the same, it's the same rate both ways, okay? Whereas and what this does is it makes our reactants and products have the same concentration when we're at equilibrium. So the dynamic steady state, the reactants and the products are at the same concentration, but we're very far from equilibrium. There are other metabolic processes that work to make sure that this, as the reactants are being consumed, they're being replenished by other reactions, and as the products are being created, they're being consumed by other reactions. So our dynamic steady state keeps, the similarity between the two is that the concentrations of the reactants and products are the same. It's just the methodology of how that happens is very different. And basically our dynamic steady state is far from an equilibrium energy state. Okay. Question 18, also not bad. I had a few people maybe not answer something in there too, or I think on the few questions I wrote, but what does this mean? So our pH is basically signifying the concentration of proton, which gives you an idea of how acidic something is. pKa, you could also say it indicates the acidity of a particular group. It's really how easily that proton is going to be extractable from that group. And any variation on that would have worked. I was just looking there. Our pKa is just, our pH is how acidic is our solution. Our pKa is how acidic is our molecule, in essence. And then the calculation here wasn't too bad. We'd use our Henderson-Hasselbalch. You've got 0.15 moles of the weak acid and 0.125 moles of the conjugate base. So you'll take your pH is equals 5.72. So 5.72 is going to equal our pKa plus the log of 0.125 over 0.150. the key to, I believe the answer works out being something like 5.8. Because we have a higher concentration of the weak acid than we do the base, the conjugate base. Yes, we should expect our pKa to be a little higher than our pH. We know our pH, we have more of the acid form, so our pH will be lower than pKa. I think maybe one person flipped the sign and got 5.64, but for the most part, this was pretty well done. We get to number 19, and better. Some people only gave me one methodology here, but if we have an affinity chromatography, we have a stationary phase that is recognizing our protein of interest. It's going to bind it, and we have to get it off of there. Okay, so basically all the other proteins ideally pass through, and our protein of interest gets bound. We could release it by adding a elution buffer that has a high concentration of our ligand. So we just outcompete it, and at least in there, if we're binding a particular part of the protein, with antibodies in there, you could do that. The other way you could do it, and most common way, is a high salt protein as well too. So the high salt will generally disrupt these hydrostatic interactions that allow it to bond and that allow it to release. There's probably like a third or fourth. I think some people said changing the pH. I'm like, okay, that could work as well too. Yep. Generally, we don't do that because we want to keep our pHs roughly the same so that we don't denature our protein or anything. And it got a little tricky. And for part B, why wouldn't these work in size exclusion chromatography? And the key here is to say, well, there's actually no binding in size exclusion chromatography. It just separates them based on size. So smaller proteins have a farther path length to travel, so they take longer to go through. Our larger proteins go right through because they can't fit in all those little cavities and tunnels and whatnot in there. So, in effect, we don't do an elution in size exclusion chromatography. We just directly separate, no need for elution. And size exclusion chromatography is really good if you have, only like a few different protein species. You've got like 4 or 5 or 6 proteins in there. You can separate them based on size. Our affinity chromatography can work from like crude extract. You might have like 1,000 different proteins in there. Ideally, you'll be able to grab just the one that you're interested in. Okay. Let me come back to that one. We got question 21 here. Okay, we're going to pick an organism and describe where it gets its energy from. So I kind of look at that one and, I'm thinking maybe, I was looking for like heterotroph versus autotroph or chemotroph versus phototroph. And, I didn't get super picky on that, looking for both of those, but some people gave me that and some people talk more about the kingdom or the phyla or went a slightly different direction with that. For part B, so I was actually not too cruel about part A, probably also because I graded this one after grading question 20. For part B, the key here was how do we get these highly unfavorable reactions to occur? We couple them with highly favorable reactions. So you basically take an energy consuming reaction and couple it with an energy producing reaction. So this might be like, say, the hydrolysis of ATP produces a lot of energy, so you can use that energy to drive the unfavorable reaction. Okay? And some people gave me some good examples for that. Okay, so what I'm going to do now, I'm going to stop screen sharing and do a little bit of board work for this last one here. All right, which means I need to close this down right here. But okay, so let's draw a picture of this. And I looked back through this and believe it or not, I was actually slightly merciful for this. Let's see here. Yeah, okay, I'm gonna stop the projector and write on the board. Okay, so what do we have here? We have a 2 amino acid oligopeptide. So we're gonna have aspartate and cysteine. Okay. The good news is, so my pK1 for aspartate, I don't need to worry about that because that's our carboxylic acid. It's going to interact with our pK2 of the cysteine, so I can kind of throw those out. All right. So if I have my pK2 for aspartate, okay, that is no, wait a minute, sorry. 9.82. And my pKr, or aspartate, is 3.86. Okay. pK1 for the cysteine, and pKr for the cysteine are 2.05 and 8. Okay, so I'm going to draw this out in a really cheating version. Okay, so let me put this in my most basic. So I'm going to say that, or sorry, my most acidic. So pH 1. Okay, we're going to make sure everything is. So my group over here is going to be NH3 plus. This is going to be COOH. This is going to be SH. This will be COOH. All right. My total charge here, +1. All right. If I raise my pH, okay, to above 2.1, all right, I'm going to take away a proton. Which proton am I going to take away? This one. So basically, this one's going to come off. So now I'll have H3N plus COOH, SH, and COO minus. My charge is 0. Okay. If I keep going, take away another proton, the next one that's going to come off is my R group for my aspartate. So I'll have H3N plus, COO minus, COO minus, and SH. Okay. So at this point, I'm at -1. Okay. I could keep going and abstract these next two away, but I'll just go to -2 and maybe -3. If I had to do it again, I don't think I'd use cysteine because that's a little funky, but you know, we end up not really needing that. So if I look at my 2 pKa's that should be on here, I need to go back and look at this. The pKa's that are coming off from here, this is 2.05. This one is 3.86. So I'm going from +1 to 0 and 0 to -1. So my PI should be 2.05 plus 3.86 divided by 2, or 2.9596, something like that. Okay. Now I need to go back and look at that because I think I put in something wrong when I first started grading them, but I'll go back and look at that and make sure. But that's how we'd actually set that up. Okay. Okay.
Is that question 20?
Yes. Yeah.
This was kind of hard because I know in class we only did one, so we did two kind of like.
Yes. Well, there are also two examples of longer ones written out there as well too in the exam 1 material. So yeah. So I think I should have pointed those out a little more strenuously because I saw a lot of people just skip this question completely, and yeah, this was definitely the hardest question.
Yeah, I didn't know how to do it. I didn't know where to
Yeah.
start.
Like I knew how to do the one, I was like, it's two, so now I'm like, and it was like 2 PK levels, 2 PK. That's what I'm doing.
Yes. So
For aspartate, what I did is 3.86 plus 9.82 divided by 2, and I got 6.84. I don't know if I get great about it with this. And cysteine, I did 2.05 plus 8 divided by 2, which is 5.025. Is that right?
well, not quite. And the whole reason is so one of the examples, and I'll bring this up next week.
Why?
Yeah. But what we're really looking is we're just looking for this balance point. So if I were to use an analogy, I got my ruler here. I've got one balance point. This is going to be my isoelectric. If I can do it here, my isoelectric point. So I might have 40 different pKa's on there.
Right.
But if I go from -1 to -2, I'm still unbalanced over here. It doesn't matter how far away. So I just want to find the 2 pKa's where I go from 0 to +1 and 0 to -1. It'll be the average of those two.
Oh, and you want, okay.
So I understood your methodology, but it was just not quite the right way. Okay, but I'll pull up that example. Actually, I think I can do that
Did we
now.
like an example about this one before the exam?
What's that?
Did we go over?
We had a very brief example, and then if I go back to,
I don't think. I don't remember.
yeah, so yeah, so there's two of them in here. So if we looked at, say, glycine, serine, aspartate, alanine, see if I can do this way. Of course, it's going to show up sideways. Actually, I can do this a little bit better. Okay. Okay. Come on, there we go. Okay, now let's rotate. Oh Lord, I got to remember how to do this in this. Rotate clockwise. There we go. Okay. And let me share the screen so you guys can. Oops. You guys can see this at home. All right. Okay, this isn't perfect. Okay. Where is the thing? Somewhere there should be. Where'd the Zoom meeting go? Really? Hope I didn't cancel everybody. Are you guys still here?
Yeah.
Yeah, we'll still.
Okay, you guys are still. I just can't find. Hold on a minute here. Let's do it this way. There we go. Everything disappears when I. Okay, so now I need to open this back up. It's a PDF. Okay. It's directly. That's weird. Okay. Share screen. There we go. Okay. Now I can do that. I can do this so you guys can see. And just take a second for that to kick up there. But again, I have a cheating shorthand method because I don't need the whole structure in there. It would actually be a good exercise to draw the whole thing out. But the key that I want to look at is my, all protonated in here. So everything's protonated. I got this OH group, my NH3+, my COOH, my COOH. I'm just kind of drawing these. This is my G, S, E, and A. That's kind of like what's cut off up here. But my glycine doesn't have an ionizable side chain. My serine does. My aspartate does. My alanine does not. So it turns out I have 4 possible ionizable protons there. If I go over here, the first one's going to come off is always my pK1. So this comes off here, and I see my charge goes from +1 to now 0. The next one that comes off is again my pK R for the side chain for aspartate. So I look at that. I don't need to go any further. I could abstract this one next and go to -2, and abstract this one next and go to -3. But all I want to find are the ones from 0 to +1 and 0 to -1. So I'm going to take the pKa's for my aspartate side chain and my alanine carboxylic acid group. Okay? So I do that and I end up with a isoelectric point of 3. Okay? The big key is recognizing that you're going to lose all but one of the pK1s because they all are participating in that peptide bond. Same way you're going to lose all but one of your pK2s because all those amino groups are participating in there. So you have your amino terminus, your carboxy terminus, and those are the only ones that you have to worry about. And usually that one's going to be the first one to come off. And it'll just depend on your side chains and your total charge. If I had like a series of lysines here, you'd have like a really positive protein. So you might have to go through like 3 or 4 or 5 steps to figure out where that comes off. Okay. But I'll let you guys kind of go over that. We're hitting our limit for the time today. I'll give you guys 2 minutes, minute and a half, all your own. Let's see here. Question. Okay, good. All right. Okay. There we go. Okay. Any other questions for anybody? Okay. Next week, we will spend Tuesday covering the antibodies and antigens and a little bit of actin-myosin. And then on Thursday, we will get into enzyme kinetics. Fun stuff. Okay.
Dr. Ebert, when is homework 2 due? There's no due date on it.
Right. I want to see how long it takes us to get into this chapter, but it'll be due the day of the exam, which is we're probably looking at the first week in October.
Okay.
Maybe that, not next week, but the Tuesday after next week. Yeah. All right. I will see you all later. So I can stop the recording.